Hi, in this video we're going to talk about mitosis. What I'd like you to do is to get a piece of paper and you're going to fold it in half this way. So you're going to take a piece of paper and fold it in half like this. So you're going to fold it just like so. So you have half a sheet of paper. Now what you're going to do is you're going to fold it in half this way. Just like so. Then you're going to take it and you're going to fold it in half this way. So you're going to fold a piece of paper just like you're going to put it into your pocket. If you folded it correctly, you should have folded it 3 times. Once this way, once this way, and once this way. When you open it up and you hold it landscape, which is this direction, you'll see that you've got 8 spaces. 1, 2, 3, 4, 5, 6, 7, 8. And it should look just like what I have on the screen. And this is where we're going to draw. So right across the top, you're going to write the word mitosis. And we are going to draw this process. Now, as we draw this process, after we're done drawing this process, you're going to be asked the question, what is mitosis? So please pay attention to what we're doing. So in this first box here in the upper left-hand corner, we're going to title this prophase. And in each of these boxes, this one and this one and this one and this one across the top, we're going to draw pictures. And in the bottom, here and here and here and here, we're going to write what's going on in those pictures up above. So what I need you to do is to draw a nice big circle in this first space here. Then what we're going to do is, and this circle is a cell. It's a eukaryotic cell. So I bet if this was a solid line, you could tell me what it is. Let me pause for a second, let you think. What is this thing in the center of a eukaryotic cell that I've just drawn? Hopefully you said it was a nucleus. Now the reason why we drew it in a dashed manner is because this nuclear envelope or nuclear membrane is dissolving. It's going away. What is also happening is we have these little T-shaped structures, and they're written in any direction. They are actually moving from, they were both together in this position here, and they're moving toward the sides of the cell. They're moving toward the poles. These are called centrioles. The centrioles are moving toward the poles. So, and then from the centrioles we have spindle fibers coming out. Spindle fibers are growing out of the centrioles. So spindle fibers will be growing out of both sides of these centrioles. But then the most important thing, the thing that we're probably going to put down here first, is the chromatin. Now, if you remember what chromatin is, let me go full screen here for just a second. So chromatin. So if we have chromatin, we have something. We have all the genetic information that's all unraveled just like this. Now if we take this and we start coiling it up and coil and coil and coil, and we get supercoils, we eventually get something that looks like this. This has been replicated and we have this. So this is a match set. These two things are part of a replicated chromosome. So this is a replicated chromosome. These are chromatids. There are 2 chromatids in a replicated chromosome. So what happens is the chromatin, which is all of the DNA wrapped around its histone proteins, will actually begin to coil. And it'll coil and coil and coil and coil and continue to coil until it eventually wraps around itself. And then after it coils, then what will happen is it forms these things called supercoils. And so the coils will continue to wrap, and they'll wrap around themselves. And what eventually happens is those supercoils are then folded back and forth and back and forth on top of each other. And then what you eventually get is you get this line, and that line is what looks like this. This is called a chromatid. when it is in the presence of its sister. These are called sister chromatids. Now we're going to talk all about these. So let me go back and share my screen with you. You guys can see what I see on the back side when I'm talking to you guys. All right, so we need to represent that chromatin coils up into chromosomes. So we're going to draw 6 chromosomes. We're going to draw one large red, one medium red, and I'm not going to draw this one complete. I'm going to draw this part over here that still is a chromatin. And then we're going to draw one small red chromosome. But I'm going to draw this part still scribbled. So we have this one has become a replicated chromosome. This one is in the process of becoming a replicated chromosome because the part of it is still as chromatin. And same with this chromosome here. So what I'm going to do is I'm going to draw 3 in blue. So in blue I need a large red. So with this one what I'm going to do is I'm going to draw part of this one not yet coiled up. And then I need a medium blue and I need a small blue. So we have several of these that have completely formed into replicated chromosomes, but we have a couple that are chromatin that is continuing to coil into replicated chromosomes. So let's list what we've got here. So let me change colors. All right. So in no particular order, we're going to list the events of prophase. So the most important event in prophase is that chromatin coil up into chromosomes. That's an H. So that's one thing that's happening. So chromatin coil up into chromosomes. So the second thing that happens, or an additional thing that happens, is, why did we draw this dashed line? Do you remember? Correct. The nuclear membrane is dissolving. It's going away because we need the nucleus to go away because we're going to do things with the chromosomes inside. We also had these T-shaped things. Those are called, do you remember? Yeah, they're called centrioles. The centrioles, that's an I, move to the poles of the cell. The poles of the cell are simply the sides of the cell. So they'll move to the opposite ends, just like the North and South Pole. These move to the move to the poles. Then we've got these spindly, fibery looking things, which is great because they're called spindle fibers. The spindle fibers form. That's a D. Let me erase that and write that again. Spindle fibers form. So we have spindle fibers form. And this is prophase. Prophase has 4 events that happen. Prophase is the first phase in mitosis. All right, so let's see if we can move this down here out of the way. All right, the second phase is called metaphase, and we're going to write that right here. And it's M-E-T-A-P-H-A-S-E. So let's go ahead and draw our cell. Are we going to draw a nucleus? I hope you said no, because over here the nuclear membrane or the nucleus disappeared. It dissolved. So now in this we have the centrioles are at the poles, so they're here. And our chromosomes, our replicated chromosomes, are going to line up. So what we need to do is we need to draw these lined up down the center of the cell. They don't need to be in any particular order. So I'm going to do my mine in this order. So I have my large, medium, and small, and then I'm going to put my other ones, which were blue. I'm going to put those, and I'll put my large one down here, put my medium one there, and put my small one there. So let's make my medium a little bit bigger. There we go. Okay. And then what happens is these spindle fibers that had formed over here, each spindle fiber is going to go from the centriole, which is at the pole, to the centromere, which I like to give you goofy associations. The centromere is merely the center of the chromosome. So if I were to use my arms and make a replicated chromosome, my wrists, where my wrists come together, is the centromere. So we go from the centriole to the centromere, from the centriole to the centromere, from the centriole to the centromere, to each one of these 6 chromosomes, just like so. Now you'll notice it connects from each side of the cell. So each centriole connects to each centromere. Now, let's describe this. So let's go back down here. Pick our color. Okay, so in metaphase, let's think about what's going on here. What's going on with the chromosomes? What did they do? How are they different here than they were there? Hopefully you've said that the chromosomes line up. Single file in the middle of the cell. There's one other thing that's going on. So talk to me about these lines here. What are they called? You're right, they're called spindle fibers. Hopefully you're actually answering when I'm pausing, because that is what will help you do well when you go to take the test. It'll help you memorize this stuff. So the centrioles are attached to the centromere That's centromeres, to the centromeres of each chromosome. Okay, so chromosomes line up single file down the middle of the cell, and the centrioles are attached to the centromeres of each chromosome by the spindle fibers. These are the two characteristics of metaphase that you need to know. So what we're going to do is we're going to pause the video at this point. What I would suggest you do is see if you can turn this paper over, see if you can draw again on another blank sheet of paper, see if you can list as many characteristics as possible, and we will be back with another video.